Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome again to the course of structural biology. We are going through the module of protein engineering. We have already talked about why protein engineering or enzyme engineering. We have talked about the method of rational design. We have talked about the utility, the application, as well as a very unique type of designing which is called de novo designing, which could even give you a protein which is not present at all, not that protein, that type of fold is not present in nature. Today we will continue from there and we will talk about something which is mimicking nature. So, the process of such type of protein engineering is called as directed evolution. As I told earlier, directed evolution is a process where we do mimic the nature. To go about the details of the method, first let us agree to an amazing fact which I talk about earlier, but you want to talk about this repeatedly. The ultimate protein designer, the ultimate biological macromolecule designer, the ultimate designer of life is Mother Nature. So, as a person, if you start thinking about the universe, about the living system going on, it is always amazing to look upon the living world, how nature being the most innovative and creative engineer of all time, how she have invented life that has flourished for billions of years under an astonishing range of conditions, variations, diversity and what not. With little bit more introspective, if you go into deep and all towards the living world, you would also be inspired by the beauty and remarkable capabilities of living system, the breathtaking range of chemical transformations they have invented everywhere, every day, every minute, every second. So many reactions, so many transformations are continuously going through, giving the life in a living mode. The complexity and the myriad roles of those products. You will be in awe of the exquisite specificity and efficiency with which nature assembles these products from simple, abundant and renewable starting materials. To maintain this for billions of years, what nature is continuously doing is development through change. There is a term, it is called evolution. Mother Nature continuously adapting the changing situation by the tool which is called evolution. So, where does this variation in chemistry come from? Because so many reactions continuously happening, so many synthesis continuously happening. It derives from enzymes, the functional proteins. As we talked from the start, that is why enzymes are so important, that is why knowing structures of enzymes is so important. So, it derives from enzyme, the DNA encoded protein catalysts that make life possible, molecular machines that perform chemistry no human creator could have matched. Equally awe-inspiring is the process by which nature created these enzyme catalysts, and in fact everything else in the biological world, the assembling, the complexity, the hierarchies, the maintenance of hierarchies, from starting from one point, how everything is regenerated, how everything is continuously operating without being stopped. The process is evolution, the grand diversity generating machine that created all life on earth starting more than 3 billion years ago. Responsible for adaptation, optimization and innovation in the living world. Evolution executes a simple algorithm of diversification and natural selection. An algorithm that works at all levels of complexity, from single protein molecule to the whole ecosystem. No comparably powerful design process exists in the world of human engineering. So, coming to the introduction to the process of directed evolution, the natural evolution of enzymes has existed since the emergence of life on earth. Genes have mutated and proteins have evolved to improve the fitness of an organism to tackle conditions in new environments. Let us say, in a very simple model, if you imagine a simple prokaryotic organism, bacteria, and in other hand, so in one side it is the bacteria, in one side it is the human. It is not difficult to understand that they have apparently no importance of time, whereas for human to live in their developed society, every minutes are important. So, the cellular function, which is simplified function distributed into different proteins in a prokaryote, in a bacteria, is not possible in human. So, we have seen that those enzymes are evolved. In some cases we see there are 3 enzymes in bacteria which comes under one complex machinery. They become efficient, they become organized, and all this is to tackle life. For thousands of years, humans have been breeding animals and plants through the selection of organisms with desired properties. For most of this time, without even knowing they were doing it, human evolved and optimized enzymes and binding proteins over many generations. So, when we will go to history, you will see that I have talked about at the initial level. So, people understood about enzymes in ancient time. They have seen that they bring an organism which is yeast and they do the brewing, they make alcohol, they use yeast to make breads and all these things. So, they understood the use of enzyme, but they could not understand the existence of those molecular machines. They actually looked at the organismic level at the ancient time, but through breeding they were trying to get better organism, but a better organism is actually a culmination of better performance of enzymes. So, through the years, on the course of making better organism, we actually have developed better enzymes, and this is what directed evolution is. So, directed evolution of enzymes and binding protein is a man-made procedure built on molecular insights which moves the evolution process into the laboratory and speeds it up. It is not that if man would not have performed it, if we do not perform, nature would not have performed it, but we have speeded it up, we have helped nature to make better organisms. The procedure relies on intended variation of protein sequences at a defined level of randomness. This is coupled to engineered screening and selection strategies. So, if you even go to the ancient time and thinking about when Gregor Johann Mendel taken the pea plants and bred them, the criteria is selection. The selection was probably been on vision, probably there was not instrumental aid to look at the quality of the peas, but it was looking at the color, the size and all those things. So, that is what engineered screening is, and ultimately that leads to selection strategies. Directed evolution is an iterative procedure which involves the identification of a starting state protein, diversification of its gene, and expression and screening strategy, re-diversification, rescreening and so on until a satisfactory performance level in terms of enzymatic activity, binding affinity or specificity is reached. So, I have talked about a big definition. I repeat it, but by splitting. So, directed evolution is an iterative procedure. What is a iterative procedure? Iterative procedure means it have a cycle. Once you do the components, you will repeat it multiple times. So, this is a iterative process which involves the identification of a starting state protein. So, to start with, you start A is a starting state of a protein or enzyme. But we know that you cannot make a change in the protein or enzyme level. So, you go to gene level or DNA level. So, the identification of a starting state protein, you have identified A, diversification of its gene, go to its gene level and you make change, then screen the expression and screening. So, you go to gene, you make change, you come up to the protein and see the expression and function, that is the screening. So, you have to develop strategies for screening, then repeating it, re-diversification, rescreening and so on until you get the desired function. So, again, directed evolution is an iterative procedure which involves the identification of a starting state protein. You identify it, go to its gene level, diversify the gene, then you screen it by expression and functional characterization and then repeat the cycle until and unless the product come with a desired property. Directed evolution of enzymes and binding proteins has become a widely used strategy in academic research as well as in the chemical and pharmaceutical industries. Directed evolution of enzymes tailors them to operate in new reaction condition, optimizes their catalytic activity towards new substrate and makes them catalyze new chemical reaction. It is like fantasy. So, you have an enzyme which convert A to B, but by the process of directed evolution, what you could do? You could enhance the speed of the reaction, you could enhance the speed of reaction. You could make new reaction condition. More interestingly, you could use C, D to their conversion material E, F, which are not related to AB, or they may be variants of AB, and makes them catalyze new chemical reaction, a complete new. So, you could make the variants or you could go for a complete new reaction. Directed evolution of enzymes has widely expanded the repertoire of useful biocatalysts. So, you have number of enzymes, but by doing directed evolution you get many different variations. So, you make diversification. The evolved enzyme offer efficient and environmentally friendly alternatives to metals and organic catalysts in chemical and biotechnological industries. Just to give you more idea, whenever you do something where a CH bond breaks and new functional bond forms that are challenging in chemistry, like if you convert benzene to phenol, apparently this is a very simple reaction, but if you want to do it chemically, it needs so many steps, it is so expensive. Now, you could use the enzyme to do that, because already we know about enzyme, we know about the specificity of the enzyme. So, it could not only make it phenol, it could also help you introducing functional group in a desired position without doing any blocking of those group. So, that is why the evolved enzymes offer efficient and environmentally friendly alternatives to metals and organic catalysts in chemical and biotechnological industries. So, directed evolution of enzymes and binding proteins in theory. What was the theory based on what the directed evolution, the mimicking of nature comes into the mind of human? As far as history says, in 1984, Manfred Eigen published a theoretical paper outlining a possible workflow for directed evolution of enzymes. Eigen noted that such optimization becomes an interesting challenge because the genotype and the phenotype are dependent on different molecule. What is genotype? Genotype is the characteristics at genetic level, and phenotype is the expression level, what you are looking, the functional level. He reasoned that finding rare improved variant in large libraries would be hard, but not impossible. Instead, he proposed the use of smaller libraries and several generations of mutagenesis and screening as a procedure that would more likely lead forward. So, he told that the variation, if you do a lot of variations, there are very less chance that you get a better enzyme than the wild type, than the original one. Instead, what he told, you make small libraries and make further changes. So, if I talk in terms of amino acids, what is suggest? Not to mutate a lot of amino acids in one go, rather mutate one or two and then do the other mutation in a different round. Why? A lot of introspectives are there from my side. One of the reason I appreciate what Manfred Eigen have told is, once you make a lot of mutations, there might be a problem of fitness. The altered protein, even if it was going towards the desired function, it was not showing you, because multiple mutation in one round might lead to the instability of the protein, because as we know, protein function in the folded form, and maintenance of the folded form is one of the most critical challenge here. But when you do one or two mutations, you check two things: one, the protein is stable or not, then the function is altered or not. Even if you have minor alteration, you could make a lot of further changes from the first generation to second generation to third generation. That is what a very good strategy. Another theoretical prediction is found in a patent that describes evolution of binding proteins through iterative diversification of libraries between selection rounds. So, kind of same thing. Directed evolution of enzyme was also briefly introduced as a possibility in an abstract of a more classical protein engineering study of enzyme optimization. This is the form of where controlled optimization by the above procedure really becomes an interesting challenge when genotype and phenotype are different molecules. So, let us therefore expand the procedure. First, you produce a mutant spectrum of self reproducing templates, separate and clone individual mutants. See, that is one. The era where we have to do the cloning and all, from the era where you are able to synthesize the gene, have changed and contributed significantly towards the progress of enzyme engineering. Now, if you are a protein engineer or enzyme engineer of this generation, you could have include anything and everything in the design. Very interestingly, you do not need even to be restricted to the 20 essential amino acids. Those were not the facility even 15 years earlier. So, here you see, produce a mutant spectrum of self reproducing template, separate and clone individual mutants, amplify the clones, express the clones, test for optimal phenotype, identify. When you get that phenotype, go for the genotype. So, you go back and then return to the initial condition, produce a mutant spectrum with a sample of optimal genotype and do next round. So, those things which were in imagination, which were in theory, start becoming in experiments, start becoming in practice. 1 decade after Eigen's theoretical work, the first experimental work appeared that described successful implementation of directed evolution of enzymes in a laboratory setting to improve enzyme function and versatility. So, what Eigen have imagined, it start coming in reality, and the first name came here is Frances H. Arnold. I hope you all know her. If you do not know, she was awarded for, in 2018, she was awarded with Nobel Prize for her contribution to developing the field of directed evolution. So, Frances Arnold reported the directed evolution of certolizin E to obtain an enzyme variant which was active in a highly unnatural environment, which is a denaturing environment at high concentration of the polar organic solvent dimethylformamide or DMF. I was actually extremely lucky in different reasons. So, in the year 2001, being into my first year of masters in the Department of Biophysics and Molecular Biology, Calcutta University, I got the opportunity to do my summer project with Professor Balram, Padmanabhan Balram in IISc, and the project I was working is an enzyme which was certolizin. I was not aware about its role in enzyme engineering, I did not know that, but what attracted me is the fact that certolizin is a protease which act. So, you know, enzyme normally works in aqueous buffer. So, in the aqueous buffer this enzyme works as a protease, whereas when it comes to the organic solvent it actually work as a ligase. So, a protease working as a ligase by changing environment. Little I know that how important this enzyme was. It was my shortcoming about the literature at that level when I was a first year student of MSc, but I was fortunate in two reasons. I worked with this enzyme and I worked with Professor Padmanabhan Balram. So, coming back here, after 4 sequential round of mutagenesis and screening in the presence of DMF, an enzyme variant with 256-fold higher activity than the wild type enzyme in 60% DMF was created. So, this was the first enzyme engineering in reality using directed evolution. In the seminal paper, Arnold has mastered the whole workflow for directed evolution of enzymes, a methodology which was continued by others in the following years. So, the protocol was having different steps. The first step was identification of a suitable starting enzyme for the chosen task, DNA sequence library construction to cover well chosen subset of sequence space. So, first you identify a suitable starting enzyme. So, certolizin was chosen, DNA sequence library construction to cover well chosen subsets of sequence space, identification of selection criteria that will lead to enhanced or new functions, and method for selection of optimized enzyme variants. One of the major challenge of directed evolution lies here. So, you get a starting material, you get the gene, you make change. Now, you made the changes and kept them in a library, you have to screen from those library to the candidates which are better. And for each and every enzyme, checking betterment is different. And if you understand, you are making many mutants. So, that is the major challenge. Then re-diversification of the genes to create new DNA sequence libraries around the sequences from the first selection to cover new subset of sequence space. And the last step, setup of selection criteria with increased stringency and so on for as many rounds as needed to reach the target level of enzyme performance. So, it is not that it is having 5 steps, it was a cycle 1 to 5 and then go to 1. But if you look at, this is the first established protocol which will give you the idea that you could actually do enzyme engineering or protein engineering using directed evolution. Each of these 5 steps has since been further developed and optimized over the years in the Arnold lab and several other labs who are working based on directed evolution. In addition to a first set of 4 combined single mutation, the first work used error prone PCR to create and re-diversify DNA sequence libraries through 3 rounds of random mutagenesis and screening to evolve certolizin E. The selection criterion was hydrolysis of the milk protein casein. Active enzyme variants were created visible halos on agar plates with casein. So, you need something which you could see or which you could differentiate. That is the first criteria of selection. Also, you need something which would make difference between one successful mutant, like sometime you make mutant they are not functional, okay. Some are functional, but you have to differentiate their activity. Now, enzymes secreted by bacterial colonies thus transfer to agar plates containing both DMF dimethyl formamide and casein, the protein, to enable identification of the most active enzyme variants in the presence of organic solvent. Then the plasmid DNA would be isolated from clones secreting an enzyme variant that produce a halo larger than those surrounding the parent enzyme and subjected to further rounds of mutagenesis. As I told, you have to differentiate, so you will only go for those colonies which are making halos larger than the wild type. The directed evolution of cetylizine E to improve its activity in a polar organic solvent was a benchmark achievement that opened the field of directed evolution of enzymes. This work became the starting point for continued technical development of the methodology of directed evolution. The field expanded towards improving and reshaping enzymes for numerous chemical reactions, old, new, leading to application of importance for research in organic synthesis as well as for the chemical and pharmaceutical industry and beyond. As I told, once you get to be empowered to change a molecule, a functional molecule created by nature, taking care of the earth, the living organisms, it is like you are playing an alternative role of Mother Nature. So, that era was opening up. Molecular insight guide library design. As we have discussed, now you understand, you have the starting protein, you go to DNA, you make change. So, you have to make a library. So, library designing is a very important part of success. It is not possible to randomize every position in an enzyme, the typical size of which is 200 to 300 amino acid residues or more. Indeed, only a small fraction of the amino acid position can be varied if the aim is a library with full sequence coverage. You understand? So, you have the entire protein, average protein is 250 amino acid. So, you range it for normal proteins from 300 to 500 amino acids. If you want to make change in each and every amino acid, just imagine the combination numbers. The reason is simple combinatorial mathematics and the quickly growing number of variants relative to the number of clones that can be handled in any laboratory setting or even using the joint capacity of all laboratories in the world. Think about, I will show you the number in the next class. You have 20 different amino acids. So, if you want to change everything, so for 100 amino acid you have 20 to the power 100, for 200 it would be 20 to the power 200. How big are those numbers? We will check it. Still, a wealth of studies makes it clear that mutation in and nearby the active site as well as more distant substitution on the enzyme surface may contribute to optimized catalytic activity. So, we start understanding if all the amino acids are equally important or the amino acids for an enzyme which is taking part in the enzyme reaction are important. So, we get a set of limited number of amino acids to change. So, that is called knowledge based selection. For a binding protein, we are trying to understand, we are trying to gain the information regarding the number of amino acid residues played critical part in binding, or other residues which helps the critical residues to be in optimal position. Arnold and co-workers have shown by many examples that library design must be based on molecular insight and knowledge based choices, of which amino acid positions to vary, combined with some element of added randomness, for example through error prone PCR. So, Arnold lab have done these things continuously, and the best part of this, they also try to elaborate their ideas to the world, so that we could also learn some strategies from them. So, what they continuously talked about, that the picking of those amino acid, the picking of the special set should be knowledge based and should be on your knowledge of molecular insight. It is not that one sunny day you come and you start doing protein engineering or enzyme engineering. You should be getting proper stepwise knowledge to know the basic rules of proteins, as well as about the particular protein which you are working, how that protein display its function. If it is an enzyme, how the amino acid residues are involved in the chemical catalysis, what type of substrates they are binding, what type of activity they have, are there loops which are involved critically, what are the movements involved, and all those they call, you need the molecular insight. But once you have chosen them, then also allow your luck, allow the nature to play by introducing some randomness through different strategies. One of them, which was popularly taken by Arnold lab, is error prone PCR. A prominent early contributor to the development and implementation of methodology for directed evolution was late William or Pim Stemmer. Stemmer introduced a DNA recombination strategy which was termed as DNA shuffling to the evolution of enzymes. This was an efficient way to propagate beneficial mutations while increasing the size of a DNA library through random fragmentation and reassembly of genes. So, mostly the concept of DNA shuffling, if I want to explain in a very simple way, take a protein called P53. P53 is a gene responsible for cancer, which generally form tetramer. Now, if you take the tetramer developing region and fuse it to an enzyme which form a monomer, this will help the enzyme to form a tetramer and increase its efficiency. Now, what I told, I know that is more a rational designing than a directed evolution, but what you see here, I cut this, I paste it here, is actually, if now you do it randomly by not knowing where you are cutting and where you are pasting, that is the DNA shuffling. I hope I make it clear. So, you have different fragments, you make pieces and then you paste randomly. What you do is DNA shuffling. Stemmer showed that the use of DNA shuffling, which is recombination of DNA from similar genes from several organisms. So, for initial experiments of DNA shuffling, what Stammer used, he brought different homologous enzymes or different enzymes with similar function from different organisms, and when you do DNA shuffling of them, it would introduce more variation than many other method and can thus improve the chances to reach a substantial activity increase in the evolved variants. In a proof of principle study, Stammer and co-workers set out to increase the activity of the enzyme beta lactamase. You know, I was almost going to tell that before coming here. So, because this, when you look at the research we do, we try to understand how the change in the beta lactamase causing emergence of new resistance. So, what we actually do, we want to understand what the nature wants to do if we invent a new drug, a new therapeutics, because starting from 1928 Alexander Fleming penicillin, a lot of beta lactam drugs are introduced in the human community. Every time we come up with new drug, nature have pushed bacteria, nature have saved bacteria by introducing a new emergent resistant gene. So, the directed evolution methodology help us to understand, and that is what Stammer do for proof in principle. He used the enzyme beta lactamase. 3 cycles of DNA shuffling and screening on plate with successively higher concentration of the antibiotic cefotaxime, which is a cephalosporin, lead to evolution of an enzyme with significantly increased activity. Gene shuffling had been reported as a means to increase variation and improve antibody affinity for a target, sometimes called affinity maturation, which is happening in the real organism, real cell, and provided early example of directed evolution of binding proteins. In these cases, the shuffled gene segment correspond to light chains or to the variable loop of heavy and light chain of immunoglobulins. So, where the variation happens, those are more important for improvement. So, how the whole process is going on? Step 1, you have the DNA which is wild type DNA. The first step is random mutation introduced. So, you see the red one. So, random mutation introduced here. Then the genes are inserted in bacteria which use them as template and produce randomly mutated enzymes. So, these are going there and randomly mutated enzymes are produced. You have taken those which have enhanced activity on which you are depending. In the step 3, the change enzyme are tested. So, now you are doing the testing. Those that are most efficient at catalyzing the desired chemical reaction are selected. Step 4, new random mutations are introduced in the gene for the selected enzyme and the cycle begins again. So, you get them and you introduce them to the next cycle. DNA shuffling staggered extension process, which is called STEP, and other method of library generation were further developed in Arnold's and Stammer's laboratories for use in the directed evolution of enzymes during the second half of the 1990s. Since the end of last century, the progressively decreasing cost of de novo synthesis of genes with degenerate codons or fully designed DNA libraries have opened a new path towards efficient and affordable production of sequence libraries with tailored diversity. So, as I told, initially you just have to depend on the existing DNA coming from nature, but now there are significant progress in the field and cost of DNA synthesis reduced a lot. So, now you need anything, you do not change, you just resynthesize. Coming to selection criteria and screening techniques. As I told, the selection criteria and screening technique are the most important part in the success of directed evolution. The selection criteria and screening techniques must be adapted for each enzyme optimization endeavor. There is no general rule, they are individualistic on every enzyme concerning to the function. Selection may be coupled to a cellular survival function. For example, the desired enzymatic activity may detoxify a compound which otherwise inhibits growth. Selection may also be coupled to a spectroscopic enzyme assay or other means of optical or ocular probing for enhanced activity. So, how it is changing, what type of change, what instrumentation, you have to optimize that. When using directed evolution to improve enzyme activity under non-native conditions such as elevated temperature or high concentration of a toxic or denaturing substance or organic solvent, the condition may have to be introduced stepwise with gradually increasing stringency of selection pressure. Otherwise, you would lose intermediates. More importantly, you probably lose everything, because you have to understand that at what level of toxicity, at what temperature, at what criteria we could make selection. At least few of them would be there. If you set in such standard where no one is selected, then your process would fail. This ensures that the enzyme template in each round has at least some rudimentary starting activity under the condition used in that round. Otherwise, you cannot do the further selection. Stepwise increase of the stress factor with intervening diversification between selection rounds makes it possible to derive successfully more effective and more tolerant enzymes or enzymes with new catalytic properties. Selection may be performed on agar plates using a filter lift assay, where you put filter and you could lift and check, or using flow cytometry in modern days. Dan Tawfik showed that directed evolution of enzyme can be set up without use of living cells. For example, using in vitro compartmentalization in water in oil emulsion droplets containing ribosomes and library of mRNA. Newer reaction conditions. The early application of directed evolution of enzymes aim to optimize the stability and the performance under new reaction conditions such as high fraction of organic solvents. Further rounds of directed evolution were added to reach a 471-fold activity increase over wild type. Many methods exist for increasing the thermostability of enzymes and other proteins. We have already talked about this. Thermostability is a very important criteria, especially when you want to take the enzyme to industry. When directed evolution of enzymes is used with an aim to increase their thermal stability, the evolutionary process may be set up as interleaved heat treatment and activity assays, or alternatively the activity assays may be performed at elevated temperature or higher temperature. In one study, the thermostability of Bacillus subtilis P. nitrobenzyl estrase was increased by over 14 degrees centigrade, which is the increase in the melting temperature. You could measure the melting temperature using circular dichroism, using differential scanning calorimetry. When you use circular dichroism, in my course I have already talked how you do the thermal characterizations. So, there are certain points where you get the spectra of alpha helix or beta sheets, and you select one point and see the change, and from the change you put the graph, you plot the graph, and you could calculate the melting temperature. So, this work showed that it is possible to improve the thermal stability of an enzyme without compromising its catalytic activity at lower temperature if both properties are constant. And as they have explained, if you see, if you have an enzyme like this and this is the residues in active site, you just make a cross around and do not touch these residues, change the other residues for thermal stability. In that way you would not affect the active site or the activity. But if you cannot make the catalytic activity retained, the evolution of one property then may come at the cost of the other, regardless of whether the two properties are inversely correlated or not correlated at all. Nature usually provides organism adapted to cold or warm environment with two different enzymes having optimal catalytic properties at low or at high temperature. Arnold showed that directed evolution can produce a single enzyme with high catalytic activity at both high temperature as well as low. So, nature usually provides one enzyme to work it at low temperature or at high temperature. So, there is a optimized, but Arnold have designed an enzyme which could work in both ways. Another directed evolution strategy relied on structural information in the form of crystallographic B factors, a measure of which regions are more or less ordered in a crystallized protein. If you remember, I have talked about that. If you remember my explanation of the coordinate files, the PDB file, you will see the thermal factor I talked about, that is called B factor. So, one of the strategy is to measure the B factors, which region the B factor is high means less ordered, where it is low means more ordered. By focusing the library of mutations to the 10 positions with highest B factor, a large increase in enzyme stability was achieved. So, what they did, they take the portion where highest B factors are there, and by making mutations they achieve higher stability. In addition to deriving improved and novel biocatalysts, directed evolution studies contribute to our general understanding of natural protein evolution process and determinants of enzyme action. If you remember, I was just talked about beta lactamase. For that enzyme which is helping bacteria to make the resistant to antibiotic, we definitely do not want by ourselves to enhance the enzyme activity, but this study helps us to understand what is our future, what type of mutations could come in future, what type of changes we could see in those enzymes. That study will give us knowledge for further drug development. Although the selection pressures operate over totally different time scales, population size, mutation rates, strength of selections, etc., Arnold and others have shown the importance of protein evolution of factors such as thermostability, the relative effect of random mutations and recombinations. They have also showed the importance of neutral drift for the evolution of protein function and correlation between the rates of protein expression and evolution, how the protein express and how its rate of evolution. Choice of starting state. Remember, we talked about this. What is your starting material? A lot is dependent on that. Arnold and co-workers have repeatedly shown that it is possible to evolve enzyme to improve their activity under new conditions in terms of solution composition, temperature and all. It is also possible to change their catalytic activity towards new substrates or a completely new reaction. This is possible as long as the enzyme that is chosen as a starting point has at least some low level of activity for the intended reaction, that is some level of catalytic promiscuity. So, this is very important. So, what they told one enzyme you know to convert A to B. the same enzyme could be involved in conversion of C to D in an efficient way if it would be easy to make directed evolution possible if this enzyme have make at least little of this conversion. An inactive scaffold is not a suitable choice. Directed evolution requires some low level of activity. So, if this enzyme is not at all doing this conversion, it would be considered as a inactive scaffold towards this transformation, and choosing an inactive scaffold is not good. So, what I was talking about, you see, if you plot the catalytic activity in the y axis and sequence space, the sequence divergence in the x axis, if the natural reaction A to B conversion is optimized, but the new reaction the C to D. So, A to B yes, C to D totally no, one condition. Another condition, A to B very good, C to D small. This is a ideal choice. You could make series of mutation from 1 to 2 to 3rd to 4th generation to come to a efficient enzyme towards the C to D reaction, but in this case this is not a good choice, this enzyme to get a efficient transformer of this reaction. So, this is the promiscuous region. The presence of this promiscuous region, how little it would be, is very much required. Even a very low activity level towards the intended reaction provides a starting state to optimize through evolution. Often just a few mutations are required to boost up the new activity. If an enzyme has a low level of activity for an intended reaction, but much higher activity for the natural one, it may be fruitful to first lower the natural activity before starting the directed evolution effort towards the new intended reaction. So, let us say this is amino acid A, this is amino acid B, this is C, this is D, this is E. If you see that for the first reaction A to B, A, B, C, D is involved, and for the other reaction C, D is involved. First you mutate these so that you could reduce this activity as much as possible. A new chemical reaction. As a recent example of this latter strategy, the activity of tryptophan synthetase from Pyrococcus furiosus was first reduced by 95% to the removal of the non-catalytic domain of the enzyme. The enzyme have another domain which is a non-catalytic domain, you remove it, that would reduce the original reaction catalysis and then engineer it. The isolated catalytic domain was subjected to 3 rounds of directed evolution to introduce new catalytic activities towards synthesis of tryptophan analogues. In a series of studies, Arnold and co-workers changed the activity of cytochrome P450 to catalyze a set of reactions for which no specific enzyme was previously available. For example, they had achieved a process called cyclopropanation, which was not done ever by any biological enzyme, because there was no biological importance of cyclopropanation, but industrially it is very important. Cytochrome P450 BM3 has a catalytic promiscuity and an ability to catalyze. with very low efficiency the cyclopropanation of styrene by ethyl diazoacetate. Much more specific and efficient enzymes were evolved, and only a small fraction 0.2% of the amino acids in the enzyme needed to be changed to optimize the new catalytic activity. These included a change of the iron ligating residue from cysteine to serine or histidine, leading to a shift in the characteristic 450 nanometer, which is because of the Soret peak comes at the 450, that is shifted to 411 nanometer, and they called the modified enzyme as cytochrome P411. Other example of reactions for which no natural enzymes have evolved are nitrogen transfer reactions. In one case, Arnold and co-workers started from a cytochrome P411, the modified one variant, and perform azide reduction about 100 times more efficiently than nitrogen transfer to sulfide. Using directed evolution, they produced an enzyme variant that instead efficiently promotes the desired nitrogen transfer process. There are several other examples of directed evolution of enzymes for carbene and nitrogen transfer reactions. Reactions with aliphatic and aromatic CH bonds are another tractable goal, because if you look at the field of organic synthesis, this is kind of a challenge to each and every synthetic chemist. So, if you could do those transformations using biocatalysis, rather to say using abiotic catalysis, like the chemical reactions which are not needed to be performed in live organism, if you could perform those reactions using your modified enzymes, because of the specificity and single step reactivity of biocatalysts, this would make revolution in the field of synthetic chemistry. Using directed evolution of cytochrome P450 monooxygenase, an enzyme was created that catalyzes intermolecular amination of benzylic CH bonds. The biocatalyst is enantioselective and lasts for up to 1300 turnovers, thereby providing an efficient biocatalyst for synthesis of valuable benzylic amines. There are many examples, but few to mention: generation of enzyme that catalyze arsenic detoxification, which is industrially important, the production of highly strained carbocycles which are actually not very stable, the switching of an enzyme from galactosidase to fucosidase, these are all done with directed evolution. Coming to few application of enzyme modification done by directed evolution. Metabolic pathway engineering is a very interesting and very demanding process nowadays. A strength of the directed evolution methodology is the ability to co-evolve enzyme in biosynthetic pathways. In one example, Arnold and co-workers evolved a multi-enzyme pathway of carotenoid production in E. coli. Her lab also showed how whole cell biocatalysts can be developed for the production of valuable chemicals by using directed evolution to enable the production of L-methionine in Escherichia coli. Biofuels: one challenge for mankind is finding suitable replacement or supplements of our fossil fuels which can be produced in a sustainable and environmentally friendly manner. This is something which we all are now focusing on, because we have enhanced population day by day, enhancing, demanding more food, demanding more resources. So, using sustainable resources are the challenges of current era. In development of biofuels, as you need to replace the fossil fuel, the target is to produce alcohol from short chain alkanes, and a leading candidate biofuel is 2-methyl-propan-1-ol, which is also known as isobutanol. Isobutanol can be produced using a biosynthetic pathway in recombinant Escherichia coli. Two enzymes in the pathway, however, require reduced nicotinamide adenine dinucleotide phosphate NADPH as a cofactor, while glycolysis, the normal metabolism during growth of E. coli, produces reduced nicotinamide adenine dinucleotide. So, NADPH, NADH, that is one of the challenge here. To resolve this obstacle, Arnold and co-workers used directed evolution to alter the cofactor dependence of the enzymes so that they instead rely on NADH, making the enzymes and thereby the organism suitable for biofuel production. In India, we also have a lot of groups working in metabolic enzyme engineering based biofuel production. One of such group is there in IIT Dehradun, Indian Institute of Petroleum, involving scientists like Dr. Debashish Ghosh, Dr. Deeptarka Dasgupta and team. Coming to new chemical bonds, carbon-carbon bond is common, but carbon-silicon bond is also common in human made chemical, but they are not present in biology. There are several reasons, not going into that, but if you could do that, that would be wonderful, but nature has not evolved enzyme that catalyze the formation of carbon-silicon bonds. However, directed evolution can be used as a strategy to ensure that such chemistry invented by humans can also be conducted by the help of enzymes. Arnold and co-workers noted that heme proteins can catalyze non-natural carbene insertion reaction. Heme protein, one of them is already talked about, cytochrome P450, but there are many other oxygenases. After screening a number of heme proteins from various organisms, they decided to use cytochrome C from Rhodothermus marinus as a starting point. This protein catalyzes the formation of carbon-silicon bonds with low efficiency, but with 97% enantiomeric excess. So, that help to select one enantioma, which is important also. A small library of variant was screened during heat treatment and in catalytic activity assays, and the best candidate was subjected to further mutagenesis and screening. The result of this work is an enzyme that catalyzes silicon carbon bond formation 40 times better than the starting enzyme and with 99% enantiomeric excess. The evolved enzyme had 15 times higher turnover number than the best non-enzyme catalyst known for the same reaction. This example shows that it is possible to expand the scope of enzyme catalyzed reaction in terms of which kinds of bonds are formed by the engineered enzyme. Other example of bonds and reactions not catalyzed by any enzyme found in nature, but for which directed evolution was used to create efficient enzymes are carbon-boron bond and enantioselective intramolecular CH amination. Enantioselectivity of enzyme. Directed evolution is an efficient way to improve the enantioselectivity of enzymes, enhancing their performance in asymmetric catalysis. The evolved enzymes are used in the production of chiral substances with high enantiomer purity. An early example of directed evolution with the aim of improving the enantioselectivity of an enzyme was reported by Machan and Bowen concerning transaminases in the catalysis of chiral amine production. This work started with an enzyme with low level of S selectivity 65% in the conversion of the ketone beta tetralone to aminotetraline, the corresponding amine. Again, a library of mutant was generated and screened for enhanced activity on S isomer, but not on the R. So, if it is on R, it was rejected in that way. The result was a biocatalyst that produced the S aminotetraline with greatly enhanced selectivity 94%, which was further improved by additional round of mutagenesis and screening. Manfred Reich and co-worker reported another early example that has led to improved enantioselectivity of lipases in ester hydrolysis. Through directed evolution via forced cycle of random mutagenesis, the selectivity factor of a bacterial lipase from Pseudomonas aeruginosa was first increased from 1.1 to 11 and then to 35 times after further diversification of the library. Directed evolution quickly made its way from the academic setting to industrial applications. Enzymes developed using directed evolution are used in industry in the production of biofuels, materials, bulk and fine chemicals, detergents, consumer products, laboratory reagents and pharmaceuticals, as well as intermediates of the pharmaceutical industry. Several of the enzymes developed in Arnold lab are used in industry. Many companies have their own scientific teams applying directed evolution strategies to improve catalyst or protein based therapeutics in terms of stability, activity, specificity and many other properties. Specific example of evolved enzymes and product are taste enhancers, drugs against diabetes and vascular plaques, as well as lipid lowering pharmaceuticals. Some enzyme produced by directed evolution are made on very large scale after confirming about their activity. This includes lipases used in detergents. Industrial chemicals are made in enormous quantities with the help of biocatalysts produced by directed evolution. So, coming to the summary about directed evolution, the use of directed evolution as a strategy for engineering novel enzymes and catabolic pathways has yet to reach its full potential. It is a kind of nascent field, and if you understand, like the first person who have kind of developed Arnold, he have just got Nobel Prize. There are certain challenges for future directed evolution that include developing workable strategies for evolution of new catalytic functions, evolving biosynthetic or degradative pathways, evolving single and new enzyme pathways for large scale chemical production, and evolving enzymes that are difficult to handle in terms of functional expression, stability and assay developments. There is a need to clearly understand what one means by a good library for evolution, and we certainly need high throughput screening tools. As I told, this is the most like critical challenge, and new assay formats to address the challenging problems to which these tools are now being applied. So, more and more automations, higher sensitivity of the screening, we could do more mutations, we could do more round of mutations within less time. So, from one generation to next generation, how less time you get, like how less time you spend to make a new set of mutation and check them, you will go to the next cycle, and more advanced cycle will give you better and better results. In vitro evolution has been reported to be a powerful and efficient tool for tuning enzyme performance in a wide range of applications. Directed evolution needs to be exploited to the maximum in certain areas such as designing novel allosteric biocatalysts, enzymes capable of chiral resolutions, that is to direct the enzyme to produce desired enantiomers. This technology needs to be used in active site protection of the chiral building blocks. Directed evolution can also be used in designing enzymes that are able to recognize certain recalcitrant entities and are able to modify them to decrease or eliminate their toxicity, and hence can have a major role in decontaminating the environment. That is one of the current day demand. One of the most critical areas, the ability of the enzymes to show activity under non-aqueous or anhydrous conditions. Laboratory evolution can help us in obtaining this type of super enzymes that can catalyze certain industrially important reactions. As a reliable catalyst improvement technology, directed evolution is expected to play a major role in removing bottlenecks of biocatalysis process technology. Directed evolution of binding proteins is an efficient way to identify variants with high affinity and selectivity for a given target. Also, it could be used to map the sequence requirements for high affinity and high selectivity protein interactions. Directed evolution of human antibodies leads to useful therapeutics. Coming to the outlook, Directed evolution of enzymes has become a highly efficient protocol for development of biocatalysts with high specificity, limited side reactions and tolerance for diverse reaction conditions. Directed evolution is a versatile and efficient path towards optimized enzymes and enzymes with novel functions. A main conclusion emerging from directed evolution research is that enzymes can indeed be tuned to catalyze new reaction and to reactions very different from the ones catalyzed by nature's own enzymes. There is ample room for optimization and redirection of enzyme function in terms of reactivity, substrate specificity and chemical reaction as well as tolerance to various reaction conditions. We are probably very far from the limit of which reaction enzyme can catalyze. There is plenty of room for further discovery and our future, it is a very bold statement, but our future is depending on expanding that repertoire. With this, I will finish today's class on directed evolution. Thank you.